All right, Peter, thank you for the introductions. And as Peter mentioned, my name is Lan Fan. I'm a project lead for dbSNP and also for this project. So today we'll be going over a brief introduction of ALFA project for those that are new to the project, and also we'll go over a little bit about the recent data release, our first release, and then we'll dive right into the tutorials on how to access the data. Then we'll conclude with some question and QA. I would encourage you to visit the project homepage. It contains a lot of the details about the project, and also in addition, it contains previous webinars, including the ALFA project introductions, and also a lot of the material that I won't be covering today. There will be a sort of webinar such as the various services that is relevant to today's talk. So this project is to provide aggregate allele frequency from dbGaP, and our goal is to provide frequency from over 1 million subjects. So I will use this as an introduction to dbGaP. This is dbGaP page. This shows all the data it has, summary data that it has. So dbGaP is the database of genotype and phenotype, and it stores studies, over thousands of studies and their results that for looking at the association between genotype and phenotype and their effect on, of course, diseases and everything else. It also had thousands of phenotype data sets and hundreds of thousands of variables and millions of subjects. And it also contains a lot of molecular data sets, including all the genotyping. And so this shows you that there are millions of subjects that have been genotyped on various platforms, including microarrays and direct sequencing from exome and whole genome. So this project is to provide all the heterogeneous data from all the different platforms and then homogenize and normalize it and report in a standard format, and then data will be integrated into a dbSNP release and then be available for various methods. So if you're already using dbSNP, there should be very little change to your standard pipeline to use this data. Okay. So just a brief summary of the data flow. So again, we select approved study from dbGaP. It goes through the QC process. Then we compute and normalize the subjects and their ancestries and assign them to a population. All the data from the various platforms convert to a standard input DCF format through a computational pipeline, where we normalize the variance, aggregate, and compute the frequency, and then provide a product. I won't go into the detail of how the variants are normalized, just to say that this has led to improvement in terms of the RS assignment and clustering, and reduced a lot of the issues that are previously, what I was saying, a previous build where we had, for instance, like duplicate RS assigned for the same variant and so on like that. This variant normalization has been published. I encourage you to read it. If you want more details, you can read it, and also there's also a webinar from a year ago that's on the project homepage. Okay. So we had our first release about a month ago, and it just started with just a modest 242 studies, and that included about 100,000 subjects, 500 billion genotypes, and the output we get is that essentially clustered down to about 500 million variants, unique variants. And although most of those already exist in dbSNP, but we also found some novel variants. Okay. So this just compared, even though from those few studies, how this compared to various projects that I've released before. And ALFA is comparable in terms of scales and scope with other projects such as NOMAD and TopMed and Exact in terms of the number of subjects and variants. Okay. So this just shows the breakdown in terms of populations from the initial release. It's still biased or primarily European, but we do have some significant numbers for some of the continental or larger populations. But our future plan is to improve to increase the number of subjects for the underrepresented population. But again, there's about, the previous release had about 100,000 subjects, but so this is just a small fraction of the available subject in dbGaP. So there's almost 2.5 million, and we already analyzed those subjects which has genotype, and I identified over almost 1.5 million subjects that could be possible candidates for best input to the ALFA project. So look forward for more data coming. We also look at comparing the data from our projects to other projects to see how they stack in terms of concordance and quality, and in terms of the ancestry, but also the variance in the populations. So here what we're looking at is we're comparing the ALFA data, minor allele frequency versus down genome. So the went down genome allele frequency is plotted on the x-axis, alpha is on the y-axis, and for each of the common population. So if they both agree in terms of for the same variant, they should just align on this 45-degree angle. And as you see, our ancestry for the populations and the variants are pretty much consistent with the 1,000 genome. And these blue lines indicate the outliers limit. So we said like pretty stringent, difference of plus or minus 0.15. And even then, we have very few variants. These are aim and informative markers that we use to interrogate or to compare, do the comparison. And there's very few outliers. And we also did the same thing with NOMAD and TopMed, and showed us good concordance between our data and these other projects. So the takeaway point is that our data, the ALFA project is consistent with other projects in terms of quality and concordance. Okay. And also the ancestry too, because if you look in that, if you compare the one-time genome and ALFA, the European population ancestry is very similar, and that we are pretty good in terms of assigning the population groups. And we also look at other variants of interest, such as the variants in ClinVar, genetic testing registry. These mostly contain the drug response markers, and also variants in dbGaP, GWAS catalog. Since any variant that has association p-value of less than 10 to the minus 5 to some particular traits of phenotype. And then we also look at variants that maybe you're interested in terms of those that have been cited in publication that may have description of phenotypes or other biological effects of the variants. As you see here, even with the small amount of subject we started with, we have pretty good coverage for most of the variants in these other resources. And if you want to get a subset of this data, it's in DCF format, you can get it from our FTP site and a supplement material. Okay. And so the alpha frequency data recorded on our dbTip RS page or RefSNP page. It's in the frequency tab. I can see here. We report the total population or the total project across all studies frequency. We report the sample size, the reference allele, and also allele frequency, and also the continental or subpopulation frequency for the, for instance, European, African, Asian, and so on. Okay. We also provide you population frequency when it's available for a variant from over hundreds of other projects that would exist in dbSNP, including found genome, exact, NOMAD, so that you can compare the data across projects. Now we'll start diving into the tutorials or the walkthrough in terms of how do you actually find the variant of interest and get to the RS page or the variant. So you can start that with searching dbSNP, and we provide a document here that provide all the supported search term, but it includes the RS number. You can search by RS numbers. You can search dbSNP by both GRC 37, GRC 38 position, gene summary, gene ID, and also other important attributes such as frequency range, ClinVar, clinical significance, and many others. So we'll go here and take a look. So this is the help page for all the supported search field and terms in dbSNP. So we provide you the field name, the aliases, some descriptions, and some of the possible values you can use for it. Then some sample queries. For instance, here you can search GRC H38 position, GRCH 37 position, ClinVar, clinical significance, and so on, and gene, and so on like that. There's also some sample query down here too. So I'll pause one second, and I'll also mention that we will be adding additional attributes specifically for alpha projects, so that will make you easier for you to search for variants that have alpha projects. Unfortunately, we didn't get it done for this webinar, but that should be in the next DBC build release in the next week or so. But because the system is so robust, I think we can get lucky, and I can still proceed with that demonstration how to find a variant with alpha specific frequency. So we'll go back to here. So here's the sample search we're going to do today. And let me just also make this full screen. So we can look for a ClinVar variant that has conflicting interpretation of pathogenicity, where perhaps maybe the frequency can inform you or provide some insight about, what the effect of the variant is. And we also want to find one where there's it's cited in publications, so that you can find these text mining or literature search or read about the phenotype and the biological effects. And then we'll just throw some other attributes like that. We want a missense variant that is clustered by validations. What this means is that it's been submitted to dbSNP multiple times. The same variant has been observed by multiple submitters. So it's essentially a low validation in dbSNP, but it's still useful. The variant may be real and that observed independently multiple times. But we'll also look for one that has no frequency from any project, so that hopefully the only frequency it has is from the alpha, okay? So this is pretty complex query. Even if you have to do this by SQL or other means, it's still pretty complex, but I'll show you how easy it is. This is to do in dbSNP. So the query that we have to generate is actually this. And don't be scared by this, because I'm going to show you how to do it using two methods, using the filter and advanced search within the Entrée search system, that you can generate this query without even touching the keyboard. So we'll proceed with that now. So I'll go ahead and click on this. So that query that I just did returned four results, right? 4 variants. So this is pretty complex query. It has a lot of Boolean joining and stuff like that. So just to show you how robust the system is. So how do you generate this query without even touching the keyboard? So if we just go to, I'll just go ahead and book a new tab. So if you're starting new, you can go to dbSNP homepage. Here's our search box. Of course, just a reminder, there's a link to the ALFA project here. Then below that, we have some example of search terms and also additional, and a link to the help page. But here you will see this filter here that will show you all the record that's available in dbSNP. So we'll just go ahead and pull that up, and then we'll use the filter to drill down the data. So see, that's fairly quick. Return, over 500 million records in no time. Okay. So here's the filter I'm talking about over on the left side here you're looking at. So we'll go ahead and check off the attribute that we're interested in. Again, ClinVar variant with conflicting interpretation of pathogenicity. So that drills down to a few thousand. But we'll take a little further. We'll go, again, we want it to be cited in publications, their RSs, so that we can get a lot of information about it. And we'll go ahead and click on missense, which doesn't change the number that much, but okay, found up 248. And then we'll want to validate by cluster, okay? So that gets you down to 248. But as you can see here, these still have frequency data as reported here. As one, we're showing you all the different projects that has low frequency. Like I said, mentioned earlier, and the next build will have one of these lines will include the alpha frequency, but right now we don't have it yet. Okay, so then that gets you down to 248. The one option you could do is also generate the query over here. You can edit this. Like I said, we're not going to touch any keyboard, and then we'll go to advanced. So this is the advanced search builder page. This will be the search box, terms. Here's the builder. And then there's some search history down here of the recent searches. So there's our initial search, we had 4. And then this is, as we drill down in the filter, it stores all the history too. Okay. So we'll go ahead and include this in the query builder, so that put that in there. But then we're going to go to this here. We're going to select not, and then exclude anything but frequency data submitted by other projects. So we'll go do it without frequency validation status. So we'll go ahead and show the index, and there's frequency, and that's it. And now what it's going to do is going to search, but exclude anything with frequency data, right? So go ahead and do the search, and there it is. Reduces down to only 4 records. You can see frequency is no longer reported for these other variants, right? And there's a little detail here to show you the variant type and all that stuff, position, allele, and of course, it's all the attributes will be selected in this sense, conflicting pathogenicity, and so on like that, validated by cluster, in publication, and so on. So we'll take a look at this one, and just RS, the first result, and see what the frequency can reveal or tell us about that variant. So if we go here, this is the detailed dbSNP RS report page. Again, the variant details up here, some summary information, public. And then over here, we have the various tabs for the different data sections. And then frequency is here, and then here's the alpha frequency data. Again, this shows you the frequency across all studies. So there's sample size, reference allele, allele frequency, and then the subpopulation. Of course, there's no frequency report from other projects, like the top meta. Okay, so this shows you that this variant allele is actually pretty common across most common population where we have data. Okay, so if you go to clinically significant, and then so what does that tell us? And because all the data is integrated here on the reference page, it's easy to try to get all the data and include the ClinVar assertions, clinical assertions. So here's the one that with the conflicting interpretation assertion of ClinVar. We'll click on that. Again, so this takes you to the ClinVar page, and it shows you about the variant detail again, some of the conditions. And then down here, you have the clinical assertion submitted by different submitters, including OMM, gene review. And then in the evidence section, you can see drill down detail what was provided in terms of how they asserted the clinical significance. So here's the OMM. We'll click on that to look. And you can see here, so here is this variant is indeed found in 40% of the patients. But then down here, it is also found in 11 or 13% of the general population, which is consistent with what ALFA reported, right? It's around in the teens, and it is a general common variant. Okay. So anyway, so that may provide you some insight into, okay, what does that mean, and so on like that. But also important, I want to get back to this page, is that this provides you a beacon essentially into dbGaP, because what this tells you that this variant exists in dbGaP, and they actually have thousands of subjects that carry this variant. So you can actually go into dbGaP and get the actual genotype if you want to investigate further, look at other variants, or interrogate the genotype of those individuals in more details, or get the phenotype of the attributes that have been described in dbGaP to see how this, if they are associated with this variant. Okay, so that is really the power of this connection between the variant and the dbGaP data, right? And here's the frequency is alpha reported here. All right, so that was that. How you do for the web search. And then next we'll move into, well, how do you do this programmatically? And we'll start using the eUtil. Again, we have a help document here specific for dbSNP, how to use the eUtil with dbSNP data. Basically, you use the same query syntax. So you can use the web to build up your query or test your query. Then you just basically just put it in your codes, and it's the same syntax, and that should work. Or you can use as a basis to dynamically generate the query, right? If you got, let's say, thousands of genes, 1,000 attributes you want to look at. So we'll go ahead and do tutorials in Jupyter Notebook. and we'll go look at that now. So dbSNP has a GitHub site, and we have a tutorial section, directory, and within that, there's a bunch of different tutorials you can walk through, and we'll go ahead and look at this one in Binder. So it takes a little while to load it. While it's loading up, I'll explain what's going on here. So we load the standard Python modules, file Python module, and we'll import in the Entree library or module. And then we'll use that to create dbSNP using eSearch, and again, just using the general same term we use for the web. And then okay, great, it's almost done. I'll go ahead and run it all just to get things started, and maybe sometimes it's, okay. So it's loading up. So here, that's loading. So here we're using the history so that we can essentially a key back that we can use to manipulate the data or retrieve it in batches. And we want 20, and this just we just send it to return 20 records. It's actually already ran it. So we do the query. We create essentially create a handle or through the eSearch. We do the query. Then we get the results. And then here's some of the parameters that return from the results that kind of help you decide what you want to do with it. So then for this query, it only returns 5 RS, and it's included here in this ID list parameter. And then there's also a query key that we'll use. And then there's some query translation. So if detail down here, in case if you zero results back, you may want to dive into the detail just to see if it's interpreted your query correctly. But anyway, so we'll get the RS list. We'll print out the RS numbers. There's an example there, and then we'll go ahead and pass those query parameters that we got back from eSearch into eFetch. And then here is download the XML for each of those records. So, and then that's that. So we could just do, so I'll go ahead and copy this just to show it to you, demonstration. And then we'll go ahead and paste it in here. All right, so I'll run it. Get the result back. And then so here, before the previous query returned 5, and we'll see what this one says it's returned 4. So the nice thing about Notebook is you can interact and play with it and experiment with it, and then even edit the codes. And then once you're done, you just go here and you go download that Python code, and you're done. You're ready to use. Okay. So that was one example. That's a simple example, and now we'll walk through a little more complicated example. So in addition to eUtils and Entree, we also have another service called the NCBI Variation Service, ATI. So this is like the Swiss Army knife or a toolbox, you can imagine, because it also does like remapping. It does conversion between different notation between HVS, BCF, RS numbers, our own internal speedy conversion. And recently we just supported GA4 HBR's variation objects. Like I said, this had been covered in a webinar a year ago, and there's a link on the bottom of the ALFA project page of that webinar, so you get more detail. I won't go into detail about that, but we did introduce 3 new functions specific for this ALFA project, and that's to retrieve the metadata about the project, the study, and the population description. And then there's one to get the retrieve all the variants within a sequence interval. And then also, of course, to get the frequency by RS numbers. And we'll walk through some of those examples now. So we have developed a bunch of tutorials at Jupyter Notebook again, including for how to do it, how to search by genes, how to retrieve by RS numbers, using VCF as an input, and so on. So we'll go there. Okay. And in addition, we have other tutorials or work notebook on how to use to do the conversion between the notation as described, the variation for service support. And there's also a module that's a wrapper around the allows to make things the code more object oriented and a little cleaner if you want to use that as a wrapper around the API. But here's all the Alpha Pacific notebooks, and they're all in binders, so you can just launch them from here. We have examples: gene, RS, and frequency, and a couple other ones here, and then one for searching for querying directly from LTP. So we'll start with this RS number, which is the simplest one. Okay, take a little second to load. Again, this is while it's loading. Again, it's loading standard modules, Python modules. So here's the call to get the frequency of RS number. Is just basically just you substitute an RS number and that's it, and then they'll get the frequency. Okay, I'll go ahead and run it all. Okay, I'll continue that. And also, in addition to that, what this does is, so here it is. So, okay, it go to return. So we retrieve as far as 16. So we return here's the object, the JSON object for that RS number. So it includes the build ID number. So if you maintain a local database, something you want to be currently up to date, you could compare the build ID. So essentially, it's just the date that a string of the date that was the build. We started the build, the alpha build. Then the result contains the chromosome number, and then position, the ref allele, and then the about project IDs for this alpha project, then the allele counts for each of the population, and the population described using biosample ID, and then the allele counts for each of those population. Okay. The next, and then also this retrieve the metadata. It's just a single call, no parameters. Retrieve the metadata, and then how to essentially just create a dictionary of the metadata, so that if you want to, instead of showing the biosample, you want to show more common terms, you can do that to translate. And then this is just some housekeeping stuff, but essentially this is the call for RS number with some error handling that you need for when you're doing doing web requests. Okay, so then it's down here. Then this I'll put it all together with the metadata and then print it out down here. Okay. Like I said before, you can pretty much interactively play with this, and then go ahead and put the new RS number in here, run it, and then you get instant results. So you can tweak and fine tune this or however you want it to for your own purpose. Okay. So we'll get out of that notebook. Then the next one is, we'll go. So this one combined both. So this is a search by gene. Directly, variation search API does not support gene search. We only support search by position. But you can get the gene position from the gene database and then use that to search the API. So here we show you how to do that by integrating both the eUtils API and the various service. Let me just run these. So you use essentially here, this function up here gets the gene location. It's using eUtils, eSummary, and the current gene database to get the gene information for the gene ID. And then it just parses the locations out and then use that location to then search, to query the variation search API to get the variant within that region. Okay, and then again, we just wrapped all this up in terms of there's some error handling and web requests. So here, using that range, we query for actually, I think it's for GP53 or yeah, GP53. So we got the range from gene database, and then we query the database. So we get over our 1,300 variants. And like I said before, these to show you the query that actually used to get the variants. There's a limit for each query of 250 variants. And so we can look at the data here. Again, it just returned as a JSON object. So you can parse that out into a more friendly format. And like I said, there's most of this code up here just to do the iterative and handling of the requests. So this is kind of nice, to show you how to integrate both eUtils and variation API. And then I think this provides more flexibility in that, so now you really can search for any genomic feature, let's say like enhancer promoters or even other variants using positions. So the other thing I want to mention is that when you query the version of this API, you're not only just getting back the existing RS variant, you're also getting the novel variants that's been discovered by ALFA. The web searches, and of course, there won't be no RS page for it. But if you want the latest, greatest data, the API and the FTP download is where you can get both the existing and novel variants. Okay. Well, I'll just go from here. All right, so we went through RS numbers, search by, if you go by RS, by gene. We're almost running out of time, so I won't go into this one, but you may want to look at this one. This one also does remapping. So it's taking input of GSC37 VCF, remap the GS38 query to the API, and we'll quickly go into this one quick. This Google Lab one, because this do the query to the FTP VCF. So the reason you want to use this is if you behind a firewall per se, and you want to actually use the FTP download or whatever reason, limitation due to web request, you can use. But this actually create our the ALFA FTP VCF. Like I mentioned before, it contains both existing RS variants and also novel variants. Quickly, just using importing the standard TabX Python module. Here we have some handling for the large requests, just housekeeping stuff. And then it just created the FTP site to get the for arrange this is chromosome 1 at this position, return the data. And it's just standard VCF format. Is a brief header, and then the standard positions, RS ID if it's available. If it's novel, it won't have an RS ID. And then ref and alt allele, and then each of the population as file sample ID, and then the allele counts. Okay. And then there's a bunch of other housekeeping function there, and then it also retrieves the metadata. And if you want to map, let's say the file sample to the before to the standard common terms, and then just put it all together down here. And then, like I said before, you can tweak around this to play around with the range of whatever. Then it'll just iterate through and just get pulling the data. Okay, so that pretty much concludes the webinar for today in terms of the tutorial. And so I just want to conclude saying that this is a growing data set. As I mentioned before, we're probably at less than % of what's available in dbGaP. So look forward to an avalanche of new data coming in the future. And we'll be updating this regularly. We'll try to get quarterly update and discover new novel variant and update population frequency data. And the other key thing is that we essentially did all the heavy lifting, taking all the heterogeneous data, normalizing it both in terms of variant, the subjects, the population, and the exchange format to use. And the other key point again is that this provides a beacon into dbGaP, and that way you can actually do authorized access, apply and get the individual genotype and their phenotypes and so on like that for further interrogation. So with that, I'll hand it back over to Peter and then follow up with questioning.
Thanks, Lon. We actually have a lot of questions, and I don't think we're going to have time to get to all of them. Sort of get to some general themes. One thing that came up a couple of times is the aggregate data from dbGaP. Those include, they include studies that include cancer patients. Is that not true?
Yes, there could be some patients in there. The nice thing with dbGaP is all that attributes have been captured. So presumably in the future, we could slice and dice the data however. So if you look at the possibility in terms of the number of attributes and the measurements, and so we could slice and dice it by cases versus control, age, sex, different cohorts, whatever, how you want, right? So the possibility is endless. So that is really the power of it. But where we're starting with just general, yes, we'll discover pathogenic variants also.
But they wouldn't, these wouldn't be somatic variants, would they, from the way these data are generated?
Well, it's hard because somatic variant calls are typically hard to identify to begin with. So there may be that, pass some filtering mechanism depending on how the study was filtered or how the assay. We have done some analysis, and we'll share that later. But we do compare some of the variants in terms of with other somatic databases to see where there may be possible somatic variant candidates. We did look at that Cosmic, I think a few other cancer sites. But
Okay.
yeah, so there could be some. But again, just the frequency may inform you, right? Because if you don't observe in 1 or 2 person out of 100,000 or a million, probably a somatic or very extremely rare mutation or rare disease or whatever. Right? Yeah.
Okay. Related to that, I think you alluded to this a minute ago. Will there be a way later on to sort of get subsets of these that wouldn't include, for example, cancer patients?
Yeah. Right now, we except this is our initial release, and it'd be great if and this resource is simply developed for our users, and the direction that we can take it will depend on what we get back, feedback we get from users. So we would like people to use this data as possible, drill down to it, tell us what is useful, what's not. And the more feedback we get from users, the more help us to prioritize in terms of what we want to take and where we want to take it, right? Millions of ways we can slice and dice this data. So we encourage users to use it. Please provide feedback, what you like to see, what you don't like, or any issues. And I think overall it will help improve for everybody else. So we really need user feedback and comments.
So you can always send on this last slide, just let me comment on that. There are some places where you can go, these URLs that are on the slide, where you can find out, more about NCBI services and so forth. But just if you want to, provide feedback, write to us at the NCBI Help Desk info. And I'm afraid we're kind of out of time, but there are a lot of questions in there that we haven't addressed fully, but I will make sure that we get them answered, and I'll put that up on the FTP site. And you'll get an email from, an automatic email from the GoToWebinar program, actually, but it will have a link to that site. And we'll get the Q&A document in there that will have the answers to the other questions that people asked. And there were a lot of good ones today. Thanks, everybody, for coming, and thanks, Lon. And I'm going to go ahead and end the webinar.